everybody. Alright, so we're going to continue the discussion we're having in a sort of asynchronous format now. And I might not go for the entire 75 minutes, but we'll see. Alright, so number 2, we're still talking about reactions like SN2 reactions, SN1, etc. What we ended with a few minutes ago was the E1 reaction and how we used it in the synthesis of amitriptyline. I'm not going to review all of that, but the main idea was that we had like a molecule, that's actually with the ketone, 2 carbons, benzene, and we essentially attached a piece with an alcohol, 1, 2, 3, and then we eliminated it through the E1 reaction with HCl, so that caused the alcohol to become a carbocation, or we protonate the alcohol, it's a good leaving group, it falls off, and we get a carbocation, and then basically this proton gets lost, and that leaves us with the final product, amitriptyline. So that's where we were. One, 2, 3 carbons, nitrogen, methyl. Okay, amitriptyline. Which is another tricyclic antidepressant drug. And I guess what's interesting here is there's a lot of drugs that have this sort of general scaffold of some sort of tricyclic backbone, and then we can make these sort of same attachments. And I'm just going to mention one second. I'm going to mention another class of molecules, and remember we're on A, which is the tricyclic antidepressants. Tricyclic antidepressants. I'm going to mention B now, which are similar molecules, are made in a similar manner, and these are antipsychotic drugs, which are also, I mean, they're psychologically active drugs, and they have a similar scaffold to the tricyclic antidepressants, even though they interact with a different receptor. Even antipsychotic and like anti-schizophrenic drugs. Like the classic example is chlorpromazine. And the molecule kind of looks like imipramine, so I'll draw it down here and then show the synthesis of it. So it's a benzene and then the nitrogen, but rather than 2 carbons, there's only 1 carbon, and actually there's a sulfur up top. I forgot about that yet. Sulfur up top, and then we have 3 carbons, 1, 2, 3, and then a nitrogen and a methyl. So this is like chlorpromazine. Has a similar structure to imipramine, which was the first one we did. It looks the same thing, except it's like carbon-carbon, you know, so the sulfur, which is kind of weird. And this inhibits different receptor. This then rather than norepinephrine reuptake, this inhibits the postsynaptic dopamine receptor. So a different neurotransmitter, dopamine receptor. And that causes this sort of antipsychotic effect. If we're going to make this using the same kind of chemistry that we've been using, well, we'd probably just attach at this point, and that's the case. So we can just make this nitrogen-carbon bond basically starting from a very simple molecule. It looks like this, the free amine, and it's also just sodium hydride, NaH, and then we use the chloro. So cool. So another class of molecules we can make using the same general synthetic approach for completely different pharmacological indications. This is used for diseases, in conditions, psychological conditions like paranoia, manic depressive disorders, conditions, etc. There's a whole bunch of uses of this in psychiatry. Okay, alright, so now off to a completely different class of molecules that we can make using kind of simple SN2 chemistry. So we're still on reactions like SN2, etc. We're going to talk now about antifungal drugs. So miconazole, econazole, and sulconazole. Sulconazole. Okay, so these are just kind of simple, small molecule drugs that are used in all sorts of fungal infections and things like that. So let's just show, and we can make them using basic organic 1 chemistry. So let's just show how we'll draw the product and then we'll show how we make it. Alright, so this is going to be for miconazole, the first one, miconazole. Got a benzene, chloro. Alright, so that's just a 5-member ring with a couple nitrogens on there. And then we have an oxygen up top with another benzene. Chloro, also kind of a simple looking molecule. This is miconazole. And it's used for, like kind of skin fungal infections, And I'll explain how it works in a second too. It's a pretty cool, simple mechanism. I think it is also called acnidazole. Probably mispronouncing that. And some of the other ones are just very common molecules that are found in like skin creams and stuff like that. Okay, so how do we make this? I mean, you can think about, when you think about like looking at a molecule like this, I mean, how could we attach the various things to this using our simple, our current reaction we're talking about is SN2 reaction. And so we could envision disconnecting, there's almost like 3 pieces, right? We got the left piece, we got the right piece, we got the top piece. And so if you use the right chemistry, the right reactions from your organic 1 toolkit, which you haven't really used in this kind of environment, you can imagine we can just sort of break this apart, and that's exactly how they do it. They can kind of attach this piece, so that you can envision just sort of attaching that piece to this piece with a proper leaving group, like a bromine or something. And then there's kind of a way to build this other piece, and they do it by attaching this piece, which is an ether, right? It's an ether linkage. This also goes back to organic 1, and the Williamson ether synthesis is just a way to build an ether, right? Build an ether from maybe an alcohol. Imagine an alcohol, attack something with a leaving group on it, this thing with a leaving group, and then it builds the ether. And then there's also this nitrogen thing will attack something with a leaving group. That's the basic idea of it. So if we go to the beginning of the synthesis, I'm just going to draw it kind of up here. What they do is they start with chloro-chloro, a pretty simple molecule, with a ketone and a bromine. Right? You can kind of see the relationship of these molecules. That ketone will eventually turn into this single bond oxygen. So double bond oxygen will be converted to a single bond oxygen, right? But with the bromine there, they're ready just to go ahead and attach this piece, right? So that's pretty cool. So all they do to get this started is they just react this with something called imidazole, which is a very simple molecule. It's essentially NH. That, right? So you can see that this and this are the same, and this is just like we're talking about SN2 reactions. So this just comes in and attacks, kicks off the leaving group, right? And what does that make? Just kind of snaps that thing on there. Nitrogen. Now it's going to look like this. Right? Yeah, that's what happens. SN2 there, What do we have to do next to get from here to there? Well, if we have a double bond O here and we have a single bond O there, we have to do something to reduce the ketone to another functional group, right? How do you do that? If we want to go from a ketone to an alcohol, let's think back from organic. Chloro, alcohol. Right? See how we go from ketone to an alcohol? How do you do that? I'm not going to reteach organic chemistry, but if we just think back a little bit, we're obviously adding a hydrogen, so there's a no H, and now there's an H right there, right? There's an H. So we have to add an H. So something that adds an H, and the thing that adds an H is called NaBH4, sodium borohydride. NaBH4. It's essentially H minus. The mechanism is really simple. It's just H minus, quotes, and it just kind of attacks that guy and kicks electrons up, and then that's how that H gets added, and that's how a ketone gets converted to an alcohol. And so now it's just a matter, back to our SN2 reaction, of just an SN2 that converts the alcohol to an ether, and that's the Williamson ether synthesis. Okay, so we gotta attach this thing to make the ether. It's actually a two-step process. It's the Williamson ether synthesis, organic 1, all organic 1 so far. And there's two steps. One step is take off the proton to make it a better nucleophile, because this has got to go attack. And then step 2 is the thing we're attacking, which is that, essentially, right? So step 1, take off the proton. Step 2, attach the piece. So it's NaH is our base. And step 2 is this thing with a leaving group on it, right? So it's essentially chlorine, chlorine, and then I think we use bromine. See how that would work? NaH takes off the proton, it makes it O-. O- is a good nucleophile. O- attacks the carbon, kicks off the Br, and now you got your ether. So that's cool. It's a pretty easy synthesis to follow. A couple steps, and now we've made miconazole. Okay, it's an antifungal drug. I'll explain how the biology of this briefly in a second. Okay, so another one, same exact thing, but it just shows the generality of this kind of chemistry, is econazole. Another drug, same kind of structure, and basically what they do is it's the same exact thing where they have this dichlorobenzene ketone with a bromine. They do the same thing, and then basically the net result is the slightly different drug. And now instead of a dichlorobenzene, it's a monochlorobenzene, a para-substituted dichlorobenzene. Okay, and then we got imidazole there. Yeah, this thing in the molecule is called imidazole. Okay, alright, so one more example. Iconazole is it's used to treat a ringworm. It's a type of fungus, a skin fungus. Oops, not ringworm, a ringworm. And other types of fungal infections. It has a couple names. The name of the molecule is called dermazole. Exostatin, other names like that. Okay, I'm going to show one more variation of this, then I'll just kind of explain the basic biology of how these work. These are really cool, kind of specific fungal treatments that don't cause problems. It's not like they hurt a human host. They would just kind of target the fungus. That's cool. So I'll show an example of another molecule, and then we'll show the biology, basically, the basic idea of the biology, which is cool. So this was sulconazole, source cell number 2, which is our reactions, mostly SN2, blah blah blah. And we're on C, which is antifungal style drugs. So sulconazole, is a similar one, but with a SUL, you might think sulfur, and that's actually true. So it's the same kind of thing, and I'm just going to abbreviate. Well, I'll draw the product, and then I'll just kind of abbreviate the synthesis. So for sulconazole, we got the dichlorobenzene, but with a sulfur. It's also got the imidazole. And the sulfur is connected to a para-chlorobenzene, like this. And that's sulconazole. Sulconazole. Okay, so they basically get to make it, I'll just kind of abbreviate, you know, we start with, or we get to this OH with the imidazole. right? That's like we showed that. That was kind of our abbreviated thing with the chlorobenzene, alcohol, imidazole type thing. And then, but to attach the sulfur is just a little bit of, it's a little different, and they do it by essentially doing an SN2 chemistry from the sulfur, kicking off a leaving group off the carbon. And sulfur is a great nucleophile, and so basically if they start with this kind of thing, they can just attack this with a leaving group on it, and that just works really well. So they do SOCl2. What does that do to the alcohol? You remember? What does SOCl2 do to the alcohol? From your organic knowledge, it converts it to a chloride. Now chloride's a good leaving group. And then they just essentially, now with a good leaving group, they just kind of attack with this sulfur thing. So I'm gonna draw it upward like this. S minus, right? S minus is a good nucleophile. Draw the little lone pair, and it just kind of goes and pops it off. So that's cool, and that's how you can make this kind of molecule. This is called Exelderm. It's also an antifungal skin cream type thing, and they all have different properties and usages pharmacologically. Okay, so the only thing I wanted to mention is, like, how do they actually work as antifungal drugs? That's a pretty reasonable question. And you know, some biology we explain more in detail than other times. So the basic idea of all these drugs is they block the biosynthesis of a specific molecule called ergosterol. And so ergosterol is a steroid. I'll show the structure in a second. It's a steroid. It's a major component of the fungal cell membrane. That's kind of cool. And it's not found. It's present in fungus. But not in animals. So it's kind of cool that these drugs block the synthesis of kind of a crucial steroid that's used in a fungus, but it's not actually found in animals. And that's how it works. And without the biosynthesis of this stuff, the fungal cell can't really you know, it's not a intact cell membrane, and then basically the fungus dies. Yeah. So the ergosterol, the steroid, is just a kind of standard steroid structure. So we draw our 6-member ring, 6-member ring, 6-member ring, 5-member ring. Standard steroid. We have a hydroxy group right there. We got an alkene right here. We got a methyl coming up towards us right there. We got a methyl coming up right here. We also got an alkene right here. So, alright, we cool so far? And then we have a little chain over here, so it's dash 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 methyl down. Actually, let me fix this part over here. Okay. Dash 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 methyl. Alkene down. Trans. And we have a methyl and a isopropyl group. Okay, so this is ergosterol. steroid. And this is that, it's basically a crucial component of the fungal cell membrane. Okay? It's also, just as a little side note, in addition to being present in fungus, it's present in other kind of pathogenic organisms, like trypanosomes. So that's the protozoa that causes the African sleeping sickness. So this is one of those tropical diseases, kind of like malaria in some ways, and anyway, I believe this may be the biosynthesis of this may be one of the drug targets for trypanosomes. Also, I'm not 100% sure this second, but yeah. Anyway, that's kind of a little bit on these imidazole-based antifungal drugs. Okay, sulconazole and the other ones. Miconazole, econazole. Okay. Okay, continuing the theme a little bit more, we were talking about these reactions like SN2, etc. A little bit of, we'll see a little bit of stereochemistry here in a bit, and remember that SN2 reactions cause inversion of stereochemistry. So let's talk now about another class of molecules, and this is going to get us a little into the realm of HIV. HIV is a huge topic, so we're going to talk a little bit about one of the HIV drugs that actually has a cool SN2 reaction in its synthesis. So this is called HIV reverse transcriptase inhibition. And I have a couple slides on reverse transcriptase that I'll show in a second also. You may have learned a little bit about this. It's one of the treatment strategies for HIV. But the drug is called zidovudine. Zidovudine or azidothiamidine. It's also called AZT. That's kind of the common name of it as a drug. It's one of the earlier HIV drugs. It has a lot of interesting chemistry behind it. It's I don't know. It's just one of the several HIV treatment options. HIV, it has multiple drug targets, protease, different enzymes, protease, integrase, reverse transcriptase, HIV fusion. I'm gonna show all of the like an overview of HIV shortly. But for now, let's just talk about it a bit. And we're going to focus on the chemistry, and then I'll go a little bit more into the biology of HIV. Okay. So as a drug, I'm going to kind of go down a little bit and draw the structure of it, and then I'm going to show the synthesis of it. So the structure of AZT is alcohol. It's kind of it's a sugar, and T is thymine. I'll show the structure of that in a bit. Thymine, and then azide. What's an azide? Do you remember what azides are? AZIDE, organic chemistry. This is a simple functional group. It's called azide. It's N triple bond, or N double bond to N double bond to N. N double bond to N double bond in. this is the drug. And it's got a very interesting biological story of how this works, right? This is azidothiamidine. You know, the naming of these things, thymidine refers to the attachment of thymine, which is the nucleotide base, attached to this furan, and you have the alcohol there. Thymine is just the base. Thymidine is this thing attached to the sugar. Okay, so this is the drug. It doesn't look that complicated, right? So let's talk about the synthesis of it, and then I'll talk about some of the biology of it. All right. So the synthesis begins, and essentially the key part of the synthesis is really, you know, what we're doing now is called retrosynthesis, where we look at the product and figure out how are we going to make this. Well, the key things are the alcohol's going to be a problem because alcohols have reactivity, so we do that protection thing. So protection will kind of mask the reactivity of the alcohol. So we'll start with a protecting group on there just to make it easy. And the other thing is the azide has a very specific stereochemistry, right? It's got the H right there, and it's got the azide down there. And it has to be installed. Azides are installed by SN2 chemistry, right? Right? You might remember that from organic. I don't know. It's like you would kind of start with this thing with a leaving group, right? This thing with a leaving group, and then we'd attach azide to it. But remember, if you're going to attach azide to this thing with a leaving group, there's stereochemistry there, right? And with a stereochemistry, and if azide attacks it, it's going to invert the stereochemistry, because that's a property of the SN2 reaction, right? So my point is that when we do this, all of this stuff, the alcohol or the leaving group's actually going to be pointing the opposite direction. It's like the leaving group's going to be upward, and then when this attacks, it kicks off the leaving group, and then now inverts the stereochemistry, right? Because that's how SN2 reactions work. So that's why I'm bringing this up as a cool example of reactions number 2 SN2, because we have stereochemical inversion. That's a whole thing about SN2 reaction, right? Remember that? Maybe not. Right? Stereochemical inversion. I mean, in a nutshell, if I have like a leaving bromine and I do a nuke attack, leaving group, now the nucleophile's on the opposite side, right? Right, that's it. That's one of the things you learned about SN2 is this nucleophile attacks, kicks off, inverts, and now the nuke's on the opposite side. Well, we're going to do that in this synthesis of AZT here. Okay. All right, so let's just jump to the beginning of the synthesis. So what they do is they start with that furan, which is the 5-member ring with the oxygen. Well, I'm just going to draw out the thymine thing now. There's nitrogen. Yeah. Double bond O. C double bond O. NH. See that? Okay, so that's the thymine thing. Thymine heterocycle. Thymine. That's what I'm abbreviating as T. Thymine heterocycle is T in my final product, right? T. Okay. And then they just, like I said, they have the alcohol going the other direction, and it'll be converted to a leaving group, and then it's going to do the SN2. So see that how we start with it going up, and then we end with it going down, so that you can see what's going to happen. And the only other funny thing is they have a protecting group on the alcohol. So the end is an alcohol, but here it's going to be protected alcohol. So they just have a protecting group on the oxygen. And so the protecting group they use is triphenyl methyl, which is PH. One you probably have not seen before unless you've had a synthesis course. It's a nice protecting group for alcohols. It's called trityl. Trityl. It is trityl alcohol protecting group. Okay. Sometimes we abbreviate it as TR. Maybe I'll do that in a second. TR. Okay. TR. Alright, TR is another abbreviation for this thing. And it's removed, because every protecting group has to be removed somehow, and this is removed with acid. Removed with acid. H3O plus, or I think maybe acetic acid, or some kind of strong acid. I think even acetic acid, which isn't the strongest acid. Alright, so that's what we're starting with. And all we got to do really is make this into a better leaving group, and then attack with azide, and then deprotect. That's all. It's actually a pretty simple synthesis. Okay, so using our organic 1 knowledge, that we know of a way to make OH a good leaving group. Because that's all we got to do is we got to make it a good leaving group. But we don't want to invert it, because sometimes if you know, let's just say SOCl2, we know that, right? SOCl2 kind of, what happens is it makes this a good leaving group, then chloride kicks off the good leaving group, and then chloride's going the wrong direction. So this is bad, because it's like just making it into a good leaving group will make it fall off, invert the chemistry, the stereochemistry. So basically the way to do this, what we want to do is make it a good leaving group but not kick it off. The way they do that, this is actually all right in the Volhard-Shore book for organic. I think it's actually VS9 even, chapter 9. Is we use this chloro SO methyl. And what this does is it basically converts us into a good leaving group, but the stereochemistry doesn't change. It doesn't invert it. So in a nutshell, this just attacks. Oxygen attacks, kicks off the chlorine, and that's it. This thing attacks this, kicks off the chlorine, and then it did not change the stereochemistry, but we've made it into a good leaving group. This thing is called mesyl. It's definitely one of the things we talked about in the Volhard-Shore book. I think it's chapter 9 or maybe 8. Maybe a little bit of it 9 or 8. Okay, so I'm going to just draw what that gives us, alright? All we do is make it a good leaving group and don't change the stereochemistry. And I'm going to abbreviate this thing trityl, TR, like we said, TR. So what do we got now? We got TRO. We got our T. And this thing is abbreviated OMS. That's the abbreviation for it. I think that's it. Yep, that's it. Okay, so that's all we did. Was we made it into a good leaving group, but not inverting it. That's because we used this kind of cool special trick here. And now we're ready to attach the azide, I think. Yeah, so we want to attack it with the azide and invert it, right? So what we want to do is make there's a thymine thing. We want the azide now, and triple bond N, and double bond N, double bond N. I always say triple bond, but I mean double bond. And we want our trityl still, OTR. Okay, so how do we do an SN2 chemistry, this SN2 reaction, to attack and kick off that good leaving group? All we got to do is throw in the nucleophile. And what's the nucleophile? We want to attack that carbon with a nucleophile to invert the chemistry and give us a N double bond N double bond N. All you got to do is throw in azide. It's lithium plus, you need a salt, N double bond N double bond N. Yeah. The charges are plus charge, plus charge, right? I got it backwards. It's minus right there. And how does this SN2 reaction work? Super easily. It just attacks, kicks it off. SN2 reaction. SN2. Cool. And that gives us the azide of AZT. Yeah? So that was easy, right? What's the last thing we got to do? What's the difference now of this to this? We just have to take off the trityl. How do we take off a trityl? We said it was acid, right? H3O. I think they just use acetic acid here, so it's just acetic acid is this stuff. And the mechanism I'm not going to go over, but it's very simple. It actually makes a carbocation. Trityl falls off as a carbocation. But anyway, that's how we make AZT. All right. It's actually not that bad, and we're using our organic one knowledge. Specifically, we're using, well, there's an alcohol protecting group, but converting the alcohol into a good leaving group without inversion, that's a little bit of a weird thing, but it was actually talked about in the Volhart-Shore book. And then we just use the azide to kick off the leaving group, and now we have a new azide, and then we just deprotect. So that's cool. Alright, so how does AZT work? I mean, like I said, HIV is a pretty complicated animal, and I'm gonna give a little bit of an overview of how HIV works and how AZT inhibits the viral gene expression, basically, or actually viral replication. Okay. All right. So the thing about it is the azide is the critical part of the molecule. All right. So if you think about how I'm going to draw a scheme here in a second of how HIV replicates its RNA and creates DNA using something called HIV reverse transcriptase. The key thing about this molecule is that it kind of resembles the growing DNA chain in HIV. Okay, so it kind of looks like, and I'm just going to draw this down here. Like if I have healthy HIV double-stranded, well, it's actually single-stranded DNA initially. It is going to look like this. All right, so we have our furan, we got our maybe a thymine or one of the other bases, and we have an oxygen, and then it kind of there's a series of other nucleotides, and okay. That's kind of the way it looks. And I'm going to write healthy viral DNA. Happy HIV, healthy viral DNA. And remember the D in DNA means deoxy. That means there's no sugar right there. Alcohol, I'm sorry. Alcohol. And so RNA has hydroxy groups, DNA doesn't. That's why we don't have an OH there, we don't have an OH there. Anyways, this is kind of, imagine this being like a replicated strand of viral DNA that's being created by HIV as it's infecting human, or now what would happen if this got incorporated into this growing DNA strand? The azide would not allow you to keep attaching to other nucleotides like this. And that's the thing, is that the azide blocks the incorporation of other nucleotides. So it's kind of like a blocking group. that's the best way to think about it, right? The azide will block the attachment of other nucleotides, so it doesn't have this kind of continuous chain. So that's what we're going to see, and we actually have cool three-dimensional images that show this. I'm going to show that in a little bit also. All right, so that's what AZT does, right? AZT basically blocks something called reverse transcriptase. I'll explain that in a second. Reverse transcriptase, and that is an enzyme that HIV uses to kind of convert its RNA into DNA, into the healthy DNA, right? And if this gets kind of attached into the growing strand, then it just stops the whole process, right? So that's why we call this an HIV reverse transcriptase inhibitor. It's a RT inhibitor, reverse transcriptase inhibitor, right? Okay, so we're cool so far. Let's see what else. Yeah, I'm gonna overview these kind of viral gene expression in a second, but before I do that, let's see. I'll say, this stuff is a little bit toxic, and it was one of the earlier HIV drugs, now kind of replaced by protease inhibitors and other types of HIV inhibitors, but it is still used, and it's often used as a combination therapy. Often in combination therapies with other HIV drugs, like protease inhibitors, and we'll talk about protease inhibition more later in the course also. So they do these cocktails where they'll administer like a reverse transcriptase inhibitor and also protease inhibitor, and they have kind of a dual effect in treating HIV. All right. Okay, so that's it for the chemistry of AZT. All right, so a little bit of biology on gene expression and RNA viruses and retroviruses. All right, so we're still kind of in this little tour based on chemistry, S&T reaction, and we're on D now, which is HIV RT inhibitors, like AZT. So let's talk briefly about just how normally genes are expressed, like in mammals. Normal gene expression. And this is also called the central dogma of molecular biology. Which you've probably heard about a lot of this in your basic biology courses. But the idea is that you would have double-stranded DNA reacting in transcription to positive sense single-stranded RNA. So DNA goes to RNA. There is a reaction that converts this, one sense of a strand of RNA to the opposite sense, ssRNA, which is so it's basically a replication process, and that's RNA replication, and that is a polymerase. It's just like this is a polymerase. This is called RNA polymerase. This is a different RNA polymerase. It's called RNA-dependent RNA polymerase. RNA dependent, because it's depending on another strand of RNA. Dependent RNA polymerase. Right? Okay, and then essentially once you have RNA, usually like a messenger RNA, translation occurs through the ribosome, and that generates proteins, right? And this is what a healthy organism, how basically genes are expressed. Okay, now if we talk about RNA viruses, which actually is not exactly what HIV is. RNA viruses, but these are a class of viruses that have a lot of different, are related to a lot of different diseases. So these include like influenza, SARS. That's another respiratory virus, MERS. So this is the severe acute respiratory virus, the Middle Eastern respiratory virus, as well as COVID-19, as well as West Nile virus, Ebola, polio, and there's other viruses. They have this general type of gene expression that is followed, that's different than normal gene expression. Okay, so just the big thing though for RNA viruses is there's no DNA intermediacy. So we don't have, it's not like we're going from RNA to DNA or something like that. There's no DNA intermediacy. These are based on RNA only. And so the idea is you have viral RNA. It can be single-stranded or double-stranded. I think these are all with a single-stranded RNA. Now they can replicate the RNA, replication of RNA. Through this RNA-dependent polymerase, just like we talked about up here. Through RNA-dependent RNA polymerase. Okay, so kind of like a normal gene expression, but we're basically just, we're not using DNA as the main genetic material. Okay, so from the viral RNA though, then there's translation. Into a viral polypeptide. And then usually a lot of these viruses, including COVID, HIV does something like this also, although HIV is not in this class here. Usually it creates a big peptide, and then there's another step, which is proteases, viral protease. And proteases are basically like little pieces of little scissors, and they snip this in various places, and then they get the individual viral proteins that are the enzymes and all the machinery and the structural proteins, etc. enzymes, and that it makes it a happy virus, and it causes disease, right? So if you can block any of these parts, you could theoretically stop diseases like SARS, MERS, COVID-19, West Nile, and these are a lot of these parts are targeted by pharmaceuticals. Okay, all right. But anyway, yeah. So it's interesting though that you would generate a viral polypeptide, and then proteases kind of clip at various places. And so one of the strategies is protease inhibitors, like we're talking about, and that would, if you block the scissors, well then it can't make these individual proteins and enzymes and this and that, and then you basically blocked the virus. Okay. All right. So now retroviruses are like a third thing we're going to talk about, which is HIV. It's a little different than this. It's close. The big thing here is there's no DNA intermediacy. But with a retrovirus like HIV, we go from RNA to DNA, and then replicate, and then it you know, it just has this DNA step in it. Okay. Okay, so let's just talk quickly about retroviruses. Then I'll show like a cartoon diagram of how these, you know, the different HIV drug targets. So retroviruses, and the key examples are HIV-1, which is the human pathogen. There's also HIV-2, which is more of a primate thing, and so the HIV-1 is the one that we usually associate with AIDS. All right. So the main way these work is you would have viral single-stranded RNA, and that's kind of like the main genomic RNA, or the you know how the genomic contents of the virus are encoded into this ssRNA, and it is reacted with this enzyme we're talking about, reverse transcriptase. And what that does is it converts the single-stranded RNA into viral single-stranded DNA. And now basically into a complementary strand of the single-stranded DNA, and then these things can sort of attach to a like a base paired unit, and then this can replicate to double-stranded DNA. Okay. All right, and then through a series of other operations, this gets integrated into the host cell nucleus and genome. So it basically gets inserted into like a human genome. And then it does the same kind of thing that the other viruses do. So eventually, you know, through a couple steps, it creates the viral polypeptide. Then you got your proteases, viral proteases, and the viral proteases are the scissors that cut the viral polypeptide, and then you get your individual viral proteins. And then through replication, and you know the replication of the virus and other, you know, effects, eventually you would get the disease AIDS. So all of these areas can be targeted by drugs. Now I'll show a little cartoon diagram of kind of what all the drug targets look like in HIV. But that's the basic idea of how retroviruses work. And compared to other RNA viruses and the normal gene expression. All right. Okay, so this is just a little cartoon diagram that I have on the various stages of HIV infection and the biochemical pathways that occur once HIV enters a host cell. And some of these steps are really nice drug targets that are used for HIV drugs. This is all available on the iLearn site in a set of PowerPoint slides that we have up there, so you can check this out. Okay, so basically how the infection occurs. The viral particle and these proteins on the surface of the viral particle, GP120 and GP41, bind to a receptor on the host cell called CD4, and essentially once they fuse, the viral RNA exits the cell. These GP120 particles are still kind of preserved on the outside of the cell and will eventually be used again as the virus replicates and is extruded from the cell. But anyway, the viral RNA is converted to double-stranded DNA using reverse transcriptase through a couple steps, and then the DNA enters the nucleus and gets integrated into the host DNA sequence. And then basically the DNA replicates, messenger RNA is produced, viral RNA is also produced and excreted, and then the capsid is the kind of the outer sheath of the viral particles. But anyway, so ribosomes create the viral proteins. The proteins get incorporated into a new capsid. The capsid contains proteases, which break down the viral polypeptide chain into all the machinery, so all the enzymes, including things like reverse transcriptase, integrase, which is another viral protein, etc. And essentially, then the virus is fully replicated and able to get extruded from the cell, and then it continues growing. Okay, so various drugs can target each of these different stages of HIV replication. So there are HIV fusion inhibitors. There's a polypeptide called Fusion that kind of interferes with the interaction of these proteins like GP120 and another one called GP41 that bind to the CD4 cell. So you can prevent HIV from ever fusing to the cell by interfering with that interaction. And that's what Fusion does. HIV reverse transcriptase, of course, as we know, is the target of that drug we were just talking about, AZT, and it kind of it just it blocks the growing the growth of the single-stranded DNA chain, which eventually gets converted to a double-stranded DNA chain. So you can block that. It has some toxicity issues. There's also drugs called HIV integrase inhibitors, and they inhibit the integration of the viral double-stranded DNA into the host DNA. And then probably I think the best class of HIV inhibitors are the protease inhibitors that block and kind of like bind to the protease in a way that prevents it from working. And then you never create the mature viral machinery that would that's kind of essential to HIV. All right. So this is just a little cartoon of some of the different drug targets. And I'm not an HIV expert, so possibly got a little bit of this wrong, but I definitely have the key drug targets identified, and I think this is a great cartoon diagram for that. So this is just a X-ray crystal structure of HIV reverse transcriptase enzyme bound to growing single-stranded DNA and is capped with AZT. So this shows what AZT does. It's kind of gets stuck in there with the azide, right? The N double bond N double bond N is the little blue spheres here, and it can't grow anymore. So it basically prevents the replication of the viral single-stranded DNA. So that's pretty cool. We'll talk more about protein crystal structures later, but this, if you know how to view protein crystal structures, you can download the PDB from the PDB database. There's some viewers, and we'll talk about that a little bit later also in the course. This is PDB code 1N6Q. I'll also show this real quickly with the program Pymol, which is a PDB viewer I like. It is something you can download, but there's a little bit of work to it. For now, I'm not going to demonstrate this, but I'll just show what Pymol looks like. It's this program, and I can kind of rotate around the protein, and there's the AZT there. Maybe for fun, I'll change the color. Let's see, color cyan. And I'll change it from spheres to sticks. Yeah, there it is. Now you can see the fine detail of the chemical structure of AZT. And the azide is the nitrogen that's shown there. And again, this pretty elegantly shows how AZT basically caps the growth and the reverse transcription from the RNA particle to single-stranded DNA. All right, I think that's it for today. I think we're out of time.